Yes, thank you, Ralph. So the next speaker is Vissia Viglietta, and I apologize with my pronunciation. Dr. Viglietta currently serves as Vice President of the Clinical Development at Wave Life Sciences, where she oversees the clinical development efforts and medical affairs activities. Dr. Viglietta has more than 12 years experience in neurology clinical development across multiple indications, including MS and Alzheimer's, and including key roles in the drug regulatory approval and phase 3 programs while at Biogen. She has spent several years focused on basic and clinical research in immunology, cellular biology at Harvard Medical School. So I hand it over.
Thank you so much, Dina. And thank you all for joining us today. I'm really delighted to be here and very excited to share with you our new clinical study with SNP3. We believe the start of the SELECT-HD study is a real testament of Wave's commitment to the patients and family facing Huntington disease, and also confirms our strong conviction to pursue a targeted and selective therapeutic approach to address the lowering of mutant huntingtin. There will be a number of forward-looking statements during this presentation. So just to start, for those of you who don't know Wave, we are a genetic medicine company that focuses on the treatment of genetically defined and very serious conditions. So in addition to HD, we are simultaneously bringing the next generation of clinical candidate to different patients with ALS and FTD for the first time addressed together with our C9orf72 targeting candidate, and also to patients with Duchenne muscular dystrophy. Similarly to the Wave3, which is our SNP3 targeting candidate, all these new molecules benefit from modification of PN backbone chemistry modification, and also leverage compelling data derived from preclinical models that help us to guide a much more robust clinical development plan. So Wave continues to pursue an allele selective approach for the treatment of HD. And this is based on the notion that both the presence of a toxic huntingtin protein and the loss of function of unhealthy huntingtin protein contributes to the disease. We also recognize the essential role of wild-type protein in supporting the health and function of the central nervous system, especially in the context of stress. We know now that the wild-type huntingtin is involved in neuronal survival, in the regulation of synaptic protein transport, in the synaptic plasticity, and also in the formation and function of cilia that are really important for the CSF flow. So we know that there is this ongoing struggle between the positive biological effects of wild-type huntingtin protein and the toxic effect of the mutant huntingtin in the CNS of people living with HD. Unfortunately, those individuals carrying an HD mutation already start at a disadvantage with approximately 50% less of wild-type protein as opposed to healthy individuals. And they keep losing ground to the disease progression, particularly if we further deplete the reservoir of wild-type protein. So for this very reason, we do believe that wild-type huntingtin preservation in the context of a disease-modifying treatment may be a key driver of efficacy due to its positive biological effects. So how are we doing this? We are able to achieve allele selectivity by targeting SNPs that are associated with the long CAG repeat that is responsible for the production of toxic mutant huntingtin protein. So we are seeking to precisely target the mutant transcript at the SNP of interest, directing the cut site to the region of the SNP, which will result in the elimination of the mutant huntingtin transcripts and consequently to a decrease of toxic mutant huntingtin, but not the wild type huntingtin. So we have a new candidate, wave 3, which is again a stereopure antisense oligonucleotide that contains this new chemistry that has the potential to really address the limitations of our first generation oligonucleotides. So first and foremost, wave 3 includes a novel PN backbone chemistry, which has demonstrated to confer higher potency, better tissue distribution, and longer durability of effect in the CNS. So unlike previous candidates, for SNP1 and 2, wave 3 also benefits from the availability of a relevant preclinical model, which enabled us to have a greater understanding of the PK-PD relationship, and this really informed our clinical development and, most importantly, our clinical starting dose. So starting with the chemistry very briefly, while our first generation molecules incorporated only PO and PS backbone linkages, wave 3 incorporates PN backbone linkages. And what this does, again, maintaining the stereopurity, this confers to the molecule a neutral charge as opposed to a negative charge. And this appears to have a profound impact on the pharmacology of the oligonucleotide in the CNS by greatly improving the cellular uptake that will result, obviously, in a better distribution of the molecule. So here, I just want to show you some of the in vitro preclinical data that we have collected with wave 3. On the left-hand side, you can see that in HD patients-derived motor neurons, wave 3 has a potent effect in decreasing mutant huntingtin, similar to that of a pan-silencing control. But differently from the pan-silencing control, though, wave 3 shows, and you can see that on the right-hand side, in vitro selectivity over different concentrations versus similar concentration of the comparator compound, which lowers both mutant and wild-type huntingtin. So we also examine the mutant huntingtin knockdown effect in the of wave 3 in the back HD transgenic mouse model, which we didn't have access to for our first generation programs. We investigated this model knowing that there were several limitations, namely the model doesn't contain the wild type huntingtin gene, and it contains multiple copies of the mutant huntingtin gene, some of which do not have the SNP3 variant, which obviously negatively affects the measurement of mutant huntingtin knockdown. Nonetheless, we have observed potent knockdown of the mutant huntingtin in the cortex and the striatum of back HD transgenic mice. And we have also seen a very durable effect out to 12 weeks that you can see in this graph. So the fact that we could show selectivity, potency, and durability of effect makes us extremely excited to start evaluating wave 3 in the clinic. And we are doing so after obviously being able to refine the pharmacological characteristics of our molecule in a number of animal models. As I said before, we explored the back HD model, and we were able to assess both pharmacokinetic and pharmacodynamic relationships following wave 3 treatment. And we achieved knockdown in relevant brain tissues. Similarly, we have measured pharmacokinetic effects in both single and multi-dose studies in non-human primates. And we have achieved there also concentration sufficient to achieve target engagement by using our back HD study as a guide and the IC50 that were derived in the back HD model. So based on this collective data on NHP and back HD, we were able to model the PK-PD relationship of wave 3 And we could derive a human equivalent dose that should attain sufficient concentration to engage mutant huntingtin transcript, both in the cortex and striatum within this, the predicted therapeutic windows. So in addition to the promising preclinical results, we are also able to leverage several learnings from the Precision HD program that we have run and immediately apply them to the Select HD trial. This includes the creation of a validated genotyping assay for SNP detection that improved the efficiency for patient identification and assessment of their eligibility. Furthermore, we developed during the Precision HD trial, actually, a novel biomarker assay to measure wild-type huntingtin levels in the CSF to confirm the selectivity of our approach. And most importantly, we benefit from experience, the amazing experience of all our clinical sites and the dedicated personnel. So very briefly, with regard to the patient identification, we have established a partnership with the Surrogen, and we were able to develop an assay to enable both at the same time the SNP genotyping and phasing with the CAG repeat expansion. So this assay was able to ensure that patients enrolled in the Select HD trial express both SNP3, obviously, on the mutant huntingtin allele. And not trivial, this assay can be actually run very rapidly, and in 1 or 2 weeks, the sites have responses. So this allowed the pre-screening of a large number of patients without any delays. As I mentioned, we have developed a measurement for wild-type huntingtin in the CSF in alignment with CHDI. And so briefly, after measuring the total huntingtin in the CSF, our wild-type assay utilizes magnetic beads that are coated with polyQ-specific antibody, which is the same antibody that we use to measure mutant huntingtin. And we use this to deplete mutant huntingtin from the CSF sample, leaving behind only the wild-type huntingtin protein that can then be quantified. Obviously, we are continuing to optimize our wild-type assay and plan to make this assay available to the broader HD community so everybody can benefit from it. So in light of all these advancements, we were able to initiate the Select HD trial to evaluate wave 3 for the treatment of early manifest HD. So Select HD is a phase 1b/2a clinical trial that consists of 2 periods, a single ascending dose and a multiple ascending dose period. We have decided, in order to optimize the evaluation of the molecule, as well as dosing levels and dosing frequency, Select HD has been designed to be adaptive, which means throughout the course of the study, an independent safety monitoring committee will guide the level of dose escalation and dose intervals. And this will help make data-driven decisions using all the available safety and PK data to really ensure an appropriate selection of next dose and next dose regimen. So for instance, as you can see here, higher single dose cohorts could start simultaneously with lower multi-dose cohorts, or one could decide just to move forward with single dose cohorts. So data will really drive dose and dose regimen. The study is planned to enroll approximately 36 patients with a confirmed HD diagnosis at early stages, and patients will have to carry a SNP3 in association with the CAG expansion. Their age will be between 18 and 60 years old, and eligible Precision HD participants can transition to Select HD after a washout period. Primary objective, obviously, will be safety and tolerability, but we will explore PK, CSF exposure of wave3, and most importantly, mutant huntingtin and wild-type huntingtin and all other biomarkers. So,
Vissia, sorry, it's Ralph. 1 more minute, please.
Absolutely, I'm done. I just want to remind you that we remain committed to an allele selective approach for HD. We are advancing wave3, which is our new generation molecule in the Select HD trial. And we have designed a very innovative adaptive trial because we want to make sure patients receive the right dose and the right interval of dosing. But most importantly, I'm super excited to announce that we have officially dosed our first patient, and we really hope to complete enrollment of the first cohort as soon as possible. Just a big thank you on behalf of WAVE to all the patients, family, advocacy organization, healthcare providers, and all the HD experts that we have collaborated with and helped us design this study and move forward with this exciting adventure. Thank you so much.
So thank you, Vissia. That was very concise and information on the study. So there are questions that have been coming in, but we will do all the questions in the panel session after everybody has presented. So I'll hand over to Ralph to announce the next speaker.